we're really excited to get going here. On behalf of myself, Katrina Walsman and Sanjay Asana, who are the co-directors of the ADEM network, and also Jennifer Dorian, our really fantastic program manager. We welcome you here today for this webinar on the health and retirement study, and especially the blood-based biomarkers. Blood-based biomarkers that Jessica Faul is going to give us an amazing overview of today. So let me start by introducing Jessica, who is a research professor in the Survey Research Center at the Institute for Social Research at the University of Michigan. She has a tremendous expertise in biomarkers. She is the co-director of the Biospecimen Lab at ISR, and also really spearheaded the addition of biomarker data to the health and retirement study. She's an expert in biological, genetic, and social science, the integration of, biological, genetic, and social science data. And she is currently the associate director of the health and retirement study. So we're delighted to have her here today. What folks should do is, as we go, feel free to put questions in the Q&A. We'll either answer them as we go, or we will definitely be answering questions at the end as well. Just a reminder that this seminar is being recorded and will be posted both on the HRS website and also linked to on our website as well, the ADEM network website. So with no further ado, Jessica, feel free to load your slides up.
Great. I just want to also just say thank you to you and to Katrina and Jennifer for helping to organize this. It's really an honor to be here. And thank everyone for attending. I can't see you, but you're out there in the web internet ether somewhere. So yeah, thanks for coming. All right, I'm going to share my screen. So can you all see that?
Yes.
Yeah. Okay, thank you. All right. Yeah, so today we're going to talk about biomarkers of Alzheimer's disease and really neurodegeneration more broadly in the health and retirement study. So it's a newer thing that we've added to the study. So we're excited to talk about it. So I'm going to go over, what the biomarkers are that we think about for this topic, how we measure them, and how well they do at prediction of cognitive functioning and dementia in a population study. So think about this sort of in contrast to a more medical or clinical-based study. So taking a step back for a minute, just to talk about biomarkers more generally, there really are different types. So when we use the word, we use it to mean a lot of different things. We could be interested in something that's investigative, so we don't really know exactly what it is, but we're trying it out in maybe an explorative setting to figure out what pathologies it might be relevant to. We could be thinking about something very diagnostic, so we really want to classify people as having the disease or not having the disease, or maybe even an exposure or not exposure at a certain point in time. We could use biomarkers to help us understand burden of disease, so really severity or extent of disease at a certain point in time. So for those kinds of things, we could have a cross-sectional study design, and that would serve us quite well. But we're also interested in biomarkers that can help us do things that are a bit more complicated. So predicting future onset of disease, these are definitely things that biomarkers that are very valuable to us because we think about them as sort of early predictors of something we can't yet measure clinically very well. Or maybe they can even help us think about the efficacy of an intervention. So, maybe it's, helping lower the risk for something, and we wanted to measure that on a blood-based, using a blood-based measure or other kind of biomarker. So for those kinds of things, we definitely have to have a longitudinal or cohort study set up. When we think about the characteristics of an ideal biomarker, really we want things that can clearly differentiate between healthy controls and those with disease. Ideally, it should detect early stages of the disease, and it should be easy to assay, highly reproducible and precise, and ideally inexpensive. So in these figures here, we have people who maybe at present day, we can measure the biomarker, and it might not be that great at distinguishing healthy people from, in this case, people with Alzheimer's disease, but maybe over time, as those two groups get a little bit more distinguished, it can do a better job. When we think about Alzheimer's disease specifically, it's one of the neurodegenerative conditions that we think about when we think about dementia. It's a major cause of dementia worldwide. And it's really tricky. It's a really difficult one to diagnose based on clinical presentation alone, because it has characteristics that are very similar to other neurodegenerative diseases. So vascular dementia, frontotemporal dementia, or dementia with Lewy body disease. So the cognitive tests that we have in a lot of our surveys really can't distinguish between these kinds of things very well. And we need to have more biological-based tests to do it, or even things like MRI. So cerebral spinal fluid has been used a lot to help kind of get at more precise try to get at more precise diagnoses for AD and AD-related dementias. And the idea is that the fluid is in direct contact with the central nervous system. And so biochemical changes in the cerebral spinal fluid are a really good target tissue for us to go looking for biomarkers. The problem is that obtaining cerebral spinal fluid needs specialized equipment, and even things like imaging that might be done as sort of a secondary part of diagnosis are really not practical ways for us to think about screening or surveying a large population in these population-based studies. So ideally, we get at something that's a little bit more easy to do. But the relationship between these CSF biomarkers and outcomes is really quite strong. And I'll talk about these different biomarkers in more detail in a minute. But what I want you to see is that for people who are cognitively normal, or people who go on to have Alzheimer's disease, there really is differentiation in these biomarkers, as early as 15 years before diagnosis. So we start to see differences in these populations quite early. And that means that these are really good targets for biomarkers that are kind of these early indicator types. And that's really what we want, because a lot of the development in drugs to treat Alzheimer's disease are targeting kind of this window. So people, before they really exhibit a lot of symptoms, they want to find them early so that they can start treatment and hopefully prolong the time that people have normal cognitive functioning or prevent disease altogether. That is why these markers are so important to us. So until the last few years, cerebral spinal fluid was required for the identification of Alzheimer's disease. But there have been scientific advances that have made the use of blood samples rather than CSF a source for these markers, reflecting brain changes related to AD and dementia. So specifically, the ones that we talk about are some that are related to AD and dementia, like amyloid plaques and tangles. So these markers are beta amyloid 42 and 40. We often think about these as a ratio. For tangles, we look at things related to p-tau. And again, I'll talk about these more in a minute. And then there's also some non-specific biomarkers related to AD pathology, like markers of neuronal damage, so neurofilament light chain, or NFL, and neuroinflammation, the marker we call GFAP. So the ones I labeled with the red A, T, and N, these are the ones that make up the ATN framework that you may have heard about or read about related to AD diagnosis. So just a little bit more about these markers. The AB42/40 ratio is one that we talk about a lot related to AD specifically. And here, a decreased ratio is a potential marker of Alzheimer's disease. We see a fairly large decrease, 40 to 60% in cerebral spinal fluid, but a lower decrease in peripheral blood. But this was one of sort of the first biomarkers related to amyloid burden that was identified. Phosphorylated tau protein is a major constituent of those neurofibrillary tangles that we often hear about related to Alzheimer's disease. And p-tau can come in lots of different forms. There can be sort of total tau, there can be p-tau 181, or like other variants of p-tau. So newer ones have come on the scene as well. And kind of on the side here, I've shown There are kind of other targets that people are looking at related to p-tau, and it's highly correlated with tau PET and CSF in the CSF tau levels. So that is another marker that is a target for AD biomarker development. One trick with this one is, and I'll talk about it a bit more later, it really requires processing and analysis quite close to the time of collection. So it's a bit tricky for population-based studies to pull off. The neurofilament light chain, or NFL, is a biomarker of neurodegeneration. Higher levels of NFL are associated with faster disease progression and brain atrophy in most neurodegenerative disorders. So this one isn't one that's very specific to AD in particular, because NFL is released into the CSF and by virtue of that, the blood, with neuronal death. So there's a lot of things that can lead to this, and it could just be sort of brain trauma generally, but it is correlated with degeneration on with neuroimaging studies, and it is correlated with plasma, NFL, and MCI and AD and ADRD cases. So it is there, it's just not specific to AD. And then GFAP is kind of a relatively newer one. Plasma GFAP has been found in people with early onset and late onset Alzheimer's disease compared to controls. Again, not so much specific to AD, but more generally neurodegeneration, because it's also found in people with a history of stroke. So when we think again about these markers, all of the markers that we're really targeting for blood-based collection are things that we see in cerebral spinal fluid, are related to the conditions that we're interested in. The problem is they're in the blood, but they're in the blood in much lower levels than they are in the cerebral spinal fluid. So one of the things that has happened that has allowed us to really look at these is that there's some newer analytic methods. So traditionally, we would look at these markers using ELISA or Luminex technologies, and newer methods like the Simoa assay using the Quantrix platform or the Mesoscale Discovery platform, or some higher sensitivity ELISAs have allowed us to actually get at the low levels that are found in the blood. So again, some of our ability to do what we can do now really happened as a result of a technology trigger. We knew what we were looking for, we just didn't have the methods to do it, but now we do. Some other challenges in developing or thinking about blood-based AD-related biomarkers are that older individuals often have multiple comorbidities, specifically cardiovascular disease and diabetes are well-established factors, risk factors for dementia, and inflammation is common to a lot of these multiple age-related diseases. So in patients that have cognitive decline in older age, they're going to have higher inflammation, but that inflammation may or may not be related to the cognitive decline they're experiencing, may be related to other comorbidities that they have. And unfortunately, some of these comorbidities can change the plasma profile for dementia. So it can be pretty complicated. So we knew a lot about the relationship between these biomarkers and specifically with cerebral spinal fluid and outcomes. And we knew a lot from clinical-based samples, but what was missing from the existing cognitive biomarker literature was replication in a representative population-based samples of older adults, including individuals from underrepresented race and ethnic groups. So when we got to a point where we felt like we could reliably measure some of these markers in blood, that's sort of when HRS feels like we can play a role. So we can kind of see how are these markers playing out in a population-based study. So we really needed to pick markers that were highly reliable and replicable in blood using plasma or serum. As I already mentioned, we really wanted things that had validated correlations or associations with Alzheimer's disease or ADRD neuropathology from cerebral spinal fluid or autopsy measures. And generally, we wanted or hoped they would be found in higher concentrations in people with cognitive impairment and AD or ADRD. So the final list of what we decided to measure, we chose based on consultation with dementia experts at the NIA Intramural Research Program. No surprise, they're the ones largely I just told you about. A beta 42-40 ratio, p-tau 181, NFL and GFAP. And we also decided to do the Olink proteomics neurology panel to measure close to 100 proteins, but I won't talk about that today. And I just want to say with respect to p-tau, because I mentioned, and those of you who know something about this topic, there are a number of different p-taus. They're all highly correlated with one another, but we chose this one because at the time, most of the research had been done on p-tau 181. So we wanted to make sure that our work was relevant to work that had been done prior to it in larger samples and more samples generally. But we're not done with challenges yet because we still have to think about how we collect the samples in a population-based study, where in our case, our respondents are spread across the entire country, and we're not asking them to come to a clinic or a central location to have their blood drawn. We're doing it in their homes, and we're shipping that blood to a central laboratory in Minnesota. So we had to be concerned about pre-analytical variation and convince ourselves that we could still measure these markers given the delayed time interval between the biospecimen collection and then processing and storage at the lab. And that can generally be anywhere from 24 to 48 hours. It's fast, but there's still a delay. It's not instantaneous. We're not getting it processed into the freezer immediately. We also were concerned about freeze-thaw effects. Generally, freezing and thawing biological samples is not your friend. It can definitely affect markers, some markers that you want to measure in your samples. So we had to prove to ourselves that we could do this, given that for us, we were probably thawing specimens that we had already collected to measure these analytes. We also wanted to think about harmonization of these biomarker measurements across different studies. I would say this is more of a concern we have currently than we did when we started this work, because I mentioned there are a lot of different platforms you can use. So our current work is figuring out how these platform results relate to each other. But in this case, we just had to pick a platform and go with it. So before we could really get started in earnest, we needed to conduct a pre-pilot to confirm that we could reliably measure the markers using the collection protocol we had already implemented in HRS, and that our results would be comparable to analytes obtained from plasma immediately extracted and analyzed, which is what most of these clinic or medically based samples do. So we conducted this pre-analytical pilot study, again, with the cooperation of our colleagues at the University of Minnesota, under the direction of Bharat Thayagarajan, who's one of the co-investigators on the health and retirement study. And so we collected sample from a lot of volunteers, and we kind of had three different arms to this. One was really to look at the stability and delayed processing. So we're looking at serum and plasma and how well those results relate or correlate with one another when we process it immediately with a delay of 24, 48, or 72 hours. We also wanted to look at within individual variation to make sure that these markers didn't change that much. We didn't think they did, but it would be really problematic had they been markers that changed, on the order of days or minutes. So what we did was we collected serum from the same volunteers at time 1, and then again in 2 weeks. We processed those immediately. And here we're just wanting to see how stable these markers are sort of in that 2-week period. And then we also used a set of tubes to test the freeze-thaw cycles and whether these particular assays were going to be robust to multiple freeze-thaw cycles. So what we found at the end of this pre-analytical pilot was that while plasma is the preferred substrate for the measurement of markers of neurodegeneration, all of the analytes for us had substantially lower levels in serum versus plasma. So we definitely saw a difference depending on which substrate we used. When we looked at the delayed processing on the immediate versus 1 to 3 days, we found decreased levels of A beta 40, 42, NFL, and GFAP with longer time. So right away, that starts to be a little bit of a concern. And we saw a more prominent decrease in plasma compared to serum. So serum was a bit more stable. Fortunately, the change in plasma was minimal for NFL and GFAP, but we really wanted to see if we could recover this A-beta 4240. And even though the absolute measure values of those two analytes decreased with time, the ratio stayed relatively stable. And since that's what we normally use, related to cognitive outcomes, we decided that we felt comfortable using the ratio and still examining the correlation with outcomes and the association with outcomes, given the delayed processing. And we would focus on that rather than the absolute measures of the individual analytes. In terms of delayed processing, it increases the level of p-tau. So the problem with p-tau is that in plasma specifically, we're normally not processing the plasma until it arrives at the lab. So that's really 24 hours after the draw. And it sort of artificially increases the amount of p-tau that's in the plasma. In contrast, serum, when we collect it in the field, we're usually spinning it down and separating the serum from the rest of the red blood cells in the field. So it's either separated in a tube after it's been centrifuged, or sometimes we even take the serum out after we spin it and we're shipping just the serum. So we're kind of separating the sample already, and that's probably helping it with its stability. So p-tau 181 was stable in serum with the delay because of that difference in the processing. The freeze-thaw cycles were definitely a factor. They could reduce the analyte concentration by 9 to 24%. But again, people stayed relatively stable in terms of their rank order. And in terms of individual differences, we didn't see any significant differences over that 2-week period. All of this is to say, it's not perfect, but we decided we felt good about those pilot results, that we would go ahead and do the assays for the HRS subsample. But we had to make some choices about how we were going to do it. So we chose to use plasma for A-beta 42 and 40. And again, we're focusing here on the ratio. We felt fine about it for NFL and GFAP. And we decided to switch to serum for the p-tau 181 assay because of the stability issue due to the delayed processing of plasma in field collection samples. Before I talk about the results, I want to talk about who we selected to be in this neuro sample pilot. So this neuro pilot that we did was selected from the 2016 Health and Retirement Study respondents born 1959 and earlier, who also participated in the 2016 venous blood study. So in the venous blood study, everybody who was in the panel in 2016 was invited to participate. It did not include the 2016 new cohort. In the end, we had just under 10,000 people participate in the VBS study. From that group, a probability sample was selected from among the respondents who were eligible for the 2016 Harmonized Cognitive Assessment Protocol, or HCAP. So this is an extra interview that happens after the HRS core interview for these selected people, and that's a much more intensive cognitive battery. We selected those people as well as people who are eligible for future HCAP. So they weren't age eligible yet, but they would age in future years. But HCAP is a random half of the HRS sample members. So here you don't have to worry so much about sample selection because of who either completed their HCAP interview or who was eligible to and may not have done it, but we still included them in this sample. So all told, the neural markers were assayed on this representative subset of HRS participants, and it was about 4,400 people that we selected. Just a bit more about the representativeness, because this question comes up a lot. Here I'm showing you the distribution, in this case, of race and ethnicity across the 2016 core, that 2016 venous blood study set of people, and the 2016 neuropilot sample. And what you can see here is it's not exactly the same, but it's quite close. So the distribution by race ethnicity is pretty identical, and this is unweighted. So this is before we've applied sample weights to these subsamples to sort of correct for any differential consent or participation. So you can see that they're really quite close before we do any correction. And for the venous blood study and this neuropilot, we have specific subsample weights to get everything back to a population representative set of estimates. Sometimes people also think that people who choose to participate in the biological portions of the study, or in this case, HCAP, are healthier than the rest of our panel sample. And here I'm showing you the unweighted percentages of hypertension and diabetes, again, across these three groups. So the 2016 core, 2016 VBS, and the 2016 neuropilot sample. And again, you can see that the distribution across the race ethnic groups for these two chronic conditions are very similar. And also with respect to things like education, here I'm just showing you for our Black respondents, the proportion of education categories, again, unweighted across these three groups. And you can see that, again, there's the samples, the core sample and the venous blood sample are almost identical. The neuro sample has slightly higher percentage of people who have college education, but really not a lot of differences here too. So these samples, even though they are subsamples of the HRS, are really don't have a substantial bias. So the subsamples are representative, even unweighted. And it's true that Black and Hispanic respondents consent and complete to these additional asks at slightly lower rates, but it's mostly random and it's not associated with things like education or health in our study. So we don't worry about these too much, and we're always monitoring it. And again, the sample weights we create adjust for any differential bias. And that's one of the strengths of having survey data. We know a lot about people. We know a lot about the people we select and don't select. And we know a lot about the people who choose to participate or don't. So we can use that in the creation of these sample weights to really correct for any differences we find. Okay, back to the neuro sample itself. It included HRS respondents over age 50. So there were about 4,400 that we selected, but 4,200 of them have weights, so they're age eligible. So the sample overlaps with the HRS 2016 measures of DNA methylation and RNA-seq. So we did a lot of other innovative measures on this 2016 sample, and we lined this set of neuropathological, neurodegenerative AD markers with those samples. So if you're interested in those kinds of measures as well, you're going to get a lot of overlap in the subsample by design. So again, the sample is inclusive of the entire HCAP 2016 sample with venous blood. And here today, I'm going to talk about sort of how these markers relate to some of our cognitive performance indicators, specifically the cognitive tests that we include in the core. I'm not going to talk about HCAP at all, that includes more. But here we have immediate and delayed word recall, serial sevens, and backward counting. So a total score ranging from 0 to 27. Using these scores, I'll also talk about how these measures relate to sort of a predicted dementia diagnosis. So using the Lango-Weir diagnostic algorithm. So that helps use those scores, it takes those scores and helps put people into buckets, the way sort of using it to figure out whether we think people are sort of in a normal cognitive functioning state, maybe have mild cognitive impairment, or we think have dementia. All of the data I'm talking about today are forthcoming. They should be released in Q3. Oh, that says of 2023, it should say 2024. And this will be an HRS sensitive health data product. So it won't be a restricted data set. A bit more about the descriptive statistics of this neuro sample. Here, this is the sample just with weights. On average, people were 68 years old. Slightly more of them were female than male. We did run these tests in different batches. So you'll see in a lot of our analyses, we adjust for batch because there are some differences across the plates that we ran. In terms of their cognitive status in 2016, you can see the vast majority of them had normal cognitive function. Only 3.6% had dementia. And then I also show you their distribution of ApoE4. So this is a genetic risk factor for dementia and Alzheimer's disease. with the number of E4 alleles you have being associated with higher risk. So we have 22% of people who have one E4 allele and only 2.3% who have two. So in terms of laboratory methods, for the NFL, GFAP, and A-beta 42 and 40 analytes, we use the Quantrix assay. This is the N4PE assay. And for p-tau 181, we also used Quantrix assay, and both of these use a Simoa machine. So again, the advantage here is that these are 1,000 times more sensitive than traditional ELISA assays, which is what enables us to find these markers at very low levels in the blood. Just so you know what's coming, in terms of the proteomic measures, we use the Olink neurology panel. So these are all proteins that have some established relationship to neurobiological processes and neurological diseases, some including Alzheimer's disease, but this is really much more exploratory. So these data will be forthcoming as well. Okay, getting into the meat of it all. So how did these all look when we did them? So we had 3 markers that were related to each other in our sample. So here, NFL related to p-tau 181 and GFAP, and then p-tau and NFL to each other. Not so much with the A-beta 42/40 ratio. And the strongest correlation was not overwhelming, but between GFAP and NFL at 0.46. So moderately correlated. In terms of the differences of these markers by age in HRS, you can see that with the exception of the A-beta 42/40 ratio, we have some really nice age-related gradients. So you see higher GFAP, higher NFL, and higher p-tau 181 in older people, which for an age-related disease like dementia or diseases like dementia is what you would expect. But again, we really didn't see much difference in the A-beta 42/40 ratio by age. When we looked at it by gender, really we only saw differences between males and females with GFAP and p-tau 181, and not so much with A-beta and NFL. And then when we looked at race and ethnicity, we saw some differences, not in A-beta 42/40, but some in p-tau and NFL, and a little bit in GFAP, but not really wildly different. And this is in contrast to some other studies that have shown very different A-beta measures, at least between whites and blacks in their samples. So here, this is the linear regression of each marker. So each individual neuropathological marker on the cognitive functioning score. So you kind of look at these separately. For us, we did not have a relationship cross-sectionally between the A-beta 42/40 ratio and cognitive function, but we did for NFL, p-tau, and GFAP in the direction that we would have expected. And that, again, is in the full sample of people, so all race ethnic groups combined. This is the same thing, but we're actually adjusting for all 4 measures at the same time. So when all the markers are considered together, NFL and GFAP still remain significant predictors of cognitive performance at that same cross-sectional point in time. When we move to a logistic regression to look at how well these markers predict dementia status, so using that Lingo-Weir algorithm, it's kind of funky to see because the odds ratios are so tiny, but GFAP is the only one that is statistically significant, but it's so small, like the odds ratio is so small, we can't really even show you. It's just very tight and just barely above 1. So none of these are really knocking it out of the park in terms of their ability to differentiate between dementia status and then MCI and normal cognitive functioning. When we look at the prediction of these markers, again, this is now looking separately within racial groups, we can see that in whites, all of the markers were related to total cognitive score in 2016. For Blacks and Hispanics, NFL definitely still hung in there. And for Hispanics, GFAP did. And then when we look again at just the ability to predict the dementia, predicted dementia, for whites, GFAP and p-tau 181 were significant, and we really didn't find much significance in the other groups. But I do want to point out for a lot of these, the estimate was the same across the groups. It was just we weren't sufficiently powered for that estimate to be statistically significant for Blacks and Hispanics. So I don't think we have enough evidence necessarily to say that the effects are different in those other, that the effects are different across race and ethnic subgroups. It's just that the effect size is small and we don't have a significantly large power to detect in the Black and Hispanic groups. When we look at longitudinal prediction, so again, we ran these samples from our 2016 collection, but by the time we had done it, it was already 2020. So we knew something about what had happened to these people in the 4 years since we collected their sample. So first we looked at the onset of dementia, so incident dementia or onset of death 2 years after the collection, and then 4. And here the analytes are standardized so that we can kind of look at their effects together. And what we found was that NFL was significantly predictive of dementia onset 2 years later. So we had an odds ratio of 1.27, and also significantly predictive of death 2 years later with an odds ratio of 1.38. We did the same thing looking at outcomes 4 years later, and we found a similar pattern with NFL being predictive of onset of dementia and death. I will say that between 2016 and 2020, about 150 people converted from normal or mild cognitive impairment to dementia. So it gives you a sense of how many people we're talking about. We also saw a relationship between p-tau 181 and death 2 and 4 years out, and with A beta 42/40 ratio and death 4 years out. I will also say that we did some additional analyses to look at whether these relationships were different in people who had ApoE4 alleles and not. And NFL was the predictor that remained significant in persons, a significant predictor of onset of dementia or death in persons with or without ApoE4. So this marker seems to be pretty robust, kind of independent of your genetic risk. I also want to say that, you know, these markers are not the only things that we collect. So a wonderful colleague of mine, Eric Klopak,
Jessica?
Yeah.
I'm going to interrupt and just, there's a few clarifying questions in the Q&A before you move on to the next one. So I'm going to throw these out there.
Okay.
I think this one is straightforward, but just clarifying that the neuro sample does in fact overlap with the HCAP.
Yes.
Yes. Okay.
I can go back to that if you want
me to.
No,
you don't. No,
okay.
You don't need to. No, I just.
Yeah, it 100% does. So every single person who was eligible for HCAP in 2016 also was put into this sample. So definitely all the people that completed HCAP and completed a blood sample are in here.
Great.
Yep.
The second question was wondering about using cognitive score. Why not use cognitive scores as a continuous outcome rather than binary? But I think I saw you present.
We did both ways. So the first set of things I reported on was cognitive performance as a continuous measure, and then we looked at it binary as well.
Great. And then this is Mark Hayward.
Hi, Mark Hayward.
He would like to know why, what are your thoughts about the small effect sizes?
About why we have them. I think that, again, I think we could see, so in the same way that we saw, I think the longitudinal collection is going to help us here. I actually think that maybe we'll see more differentiation over time. We already saw it sort of 2 and 4 years out where we had stronger odds ratios sort of at those times. And we see that also, in those early graphs the figures that I showed, the relationship with CSF. So the difference between people who go on to become cases and controls gets bigger over time. So I'm thinking that is some of it. And like I said, I think the effect size, the differences across race and ethnic groups, at least in our sample, are not very big. So I don't think yet, we haven't seen a lot of evidence that there's a lot of sort of race or ethnic differences in at least the effect size of these markers. I know that only partially answered his question, but the best I have for him now.
He said it's very helpful.
Oh, thanks. I'll be happy to chat about it with him later. Anyway, what I also wanted to show was that Eric's work actually took not all, but 80 different markers in the health and retirement study that we have, and used a machine learning model to try and figure out which ones were the ones that were the most highly predictive of cognitive function in the HRS. And what you can see is all 4 of these markers are sitting in a list of the top 16. So even though they have small effect sizes, and even though they're not always significant predictors, either cross-sectionally or in different race ethnic groups, in these kinds of models where we're kind of throwing everything in there and really saying, pick out the ones that are the most predictive, all 4 of them are in here. What I also want to point out, though, is that epigenetic clocks are also in here, interestingly. So pheno age, Grimm age, and Dunedin pace are all epigenetic clock measures that are predictive algorithms designed to use methylation to predict sort of phenotypic outcomes in older age or death. And they are quite good predictors of cognitive function as well. But notice some other things are on here that we would expect, like the educational attainment polygenic score, and some things on here that I think we were surprised to see, like peak flow. And maybe not surprised to see it in total, but surprised to see it at the top. But again, this is just to remind you that these markers are predictive, and they sit within a suite of 80 plus other markers that we have that you can use in your models, to think about how these things relate to cognitive decline and function later in life. So this work is under review right now. So in conclusion, the take-home messages here are that biomarkers can be measured in population studies with appropriate protocols. It's not exactly the same as we would do in a clinical sample, and you've got a few limitations, but it can be done. The markers are moderately correlated with one another, but not perfectly, so they are getting at some different things. And the markers also relate to cognitive outcomes. We saw very few race or ethnic differences, and again, this lack of significance is likely due to small effect sizes and insufficient power. And the biomarker story here is likely to be complex. Dementia is a messy phenotype. You saw on that slide where in the Venn diagram, we had all these overlapping circles. It's difficult to diagnose clinically and get that diagnosed really specific, which means it's going to be hard for us to find very specific biomarkers if we don't have a really good kind of end phenotype outcome to train it on. But maybe these individual markers can help us distinguish between different types of dementias. So if we decide that A beta is required for Alzheimer's disease, then we can use our biomarkers maybe to help distinguish people into buckets of the different kinds of dementias we think they have. And again, like I said, examining longitudinal change in biomarkers will be very useful for us, both to see how quickly they change and how those changes are related to later outcomes. And that is what I have for you today. Thank you. I just want to acknowledge funding from the National Institute on Aging for the Health and Retirement Study generally, but also specifically for this pilot study, and also funding from the Alzheimer's Association.
Great, thank you, Jessica. We do have some questions in here, so I'm gonna kind of work my way through them.
Rattle them off.
Rattle them off. Okay, so they're not exactly in order, but I should be able to answer them all. What was the ML algorithm used for feature selection in this study?
Oh, in the ERIC study, I'll have to go back and look, but I think it was some sort of lasso regression.
Okay, so what I will say is this is being recorded live. So for questions and here, note folks that you can come back to this because it'll be either on our website, on the EDEM network website, or on the HRS website. Jessica has also generously offered to answer some additional questions that weren't answered basically after this meeting. And so we will be able to post, for example, the answer to this question with some certainty after.
Yes, so where I don't know specifically, in the written version, I'll get it 100% right.
Okay, great, perfect. We're gonna ask you to go back to your slides again.
Sure, yeah.
The intercorrelation slide, if you could just put that up. But you can just put it up. Someone was asking to see it again. There isn't really a question, at least at this point.
Okay. The correlation between all the markers, this one.
I believe that's right, the intercorrelation slide.
Yep.
Okay, perfect. Could you repeat how many people got dementia at the 2 and year 4 follow-ups?
I know by year 4, it was about 153, I think, was our 4-year incidence.
Okay, great. And do you remember at the 2-year by any chance?
No, but I will have that in the written version.
Perfect, thank you. This is a comment, but I'd be curious to hear what you had to say of it. I'm stunned by the large effect of peak flow, a relatively simple measure. Yeah, do you have thoughts about that, Jessica?
I was as well. I don't, so here I don't think that I don't know. I don't think that there is a causal mechanism. So this is one of those things that I think might be correlated, but not mechanistic. But like I said, Eric and another colleague of mine were sort of doing independent things, and both came up with peak flow being this pretty strong indicator. So it could just be that it's like another marker that really changes with age and that is correlated. But it could also be that it's related to cognitive decline. Remember, COVID is in here. So COVID is happening in 2020, and that's going to affect also what we see. So it could be that that's playing out as well. I think we won't know until we have more waves with more measures.
Okay, and so I think maybe signal good sign for data analysis in HRS, maybe a good question to be looking at.
Yeah, definitely.
Okay, no more questions about the ML algorithm, particularly whether it's.
You'll have to wait for Eric's paper to come out.
You can come, yeah, that'll come up.
I put it in there as a teaser and to remind us that, these are very specifically targeted, right? Like, we went after these specifically because of their very strong relationships with CSF and Alzheimer's disease. But, there are other markers out there too. But again, remember what I said before about the difficulty with markers related to dementia is that there's a lot of other comorbid processes that are going on. So, association isn't causation. So remember that. But, we're really trying to put out a lot of markers here. And, so there's really a lot to choose from, if you really want to start diving deep into what we've got. So.
Okay, great. Since AD biomarkers are not routine to assay in labs, basically someone was wondering if you could give a rough cost estimate for the plasma biomarker assays.
Oh, I'm gonna have to do that one in the written version. I'll have to go back and look, but the prices for biomarkers tend to drop over time. So when technologies are very new, they can cost a lot, and then they go down, but I will put in the written version approximately how much we paid for these.
Okay, a question about whether APOE4 was related to AB and plasma.
So I didn't show it, but we did actually look at the correlation between these markers and those APOE alleles. And from what I'm remembering, there was actually not a lot of correlation. And in fact, it wasn't with A beta at all. It was with NFL. So I think NFL was the one that had correlation with APOE E4 alleles, and the other ones didn't.
Okay, great, thank you.
Mm-hmm.
A question about whether those small effect sizes might had something to do with the scale of the measures.
Yeah, I can see that.
Okay. And I think they're sort of asking, you know what, I'm going to put the rest of this question you can follow up with later, because I think it's just really some suggestions about how you might redo analyses.
Yeah. I mean, I will also say that HRS is largely funded to collect and put this data out.
Yes, exactly.
And this was really meant to be sort of a teaser of what is out there. You know, we do this kind of really basic analysis to show ourselves whether these worked at all. It's kind of a mental check QC process. We really aren't here to exhaust all the analytic possibilities. That is why we put the data out into the public domain, because we hope you will do that. So I'm happy to have suggestions, but I'm also happy if you as users download these data and work on them yourself.
Okay, so in light of that, I'm going to follow up with these questions. Are these data already available for researchers? How can we get access to it?
Nope, they will be released at the end of this summer. So we're just finalizing the documentation and the sample weights, and then they will be put out on the HRS website as a sensitive health data product.
So there are instructions on the website about how to actually access these data.
There will be when it gets there, but you can look right now. If you're not a registered HRS data user, you can go on the web and do that immediately, and you would have immediate access to all of our public data. The sensitive health measures are things like biomarkers. We kind of put that, it's one extra step you have to do, just to sign a form to get access to those. It usually happens within 48 hours. Then we have restricted data as well, but that's a different process, and these won't be restricted data. These will be sensitive health products, and as soon as they're up, you'll have access to them.
Okay, so again, because there's a bunch of questions about this, end of the summer.
End of the summer is our target. Yep. And if you're a registered user, I think you can sign up for email notifications from us. There will be a big data announcement when these data are released. So if you're getting emails from us, you'll be the first to know.
Are new waves of biomarkers slated to be collected soon? I'm going to ask this quite separately. New waves of blood, and then when are there?
So we are collecting blood most every wave now. The idea is that everybody gets it once every 6 years. So we just did the follow-up on this sample in 2022. We're out in the field again to do it in 24 and do it again in 26 and 28. 2022 was another HCAP year. So we have funding to measure all of these markers on that HCAP sample, and I think an even larger sample than that, possibly everyone we collected in 2022. So we definitely have intention both to continue venous blood collection generally, and more specifically to measure these markers going forward.
Okay, and so these are related questions, actually.
Sure.
There's sort of a question here about the ways that HCAP could be used in longitudinal mediation models. Basically, it sounds like these will become longitudinal databases over time.
So again, every time we do HCAP, there will be a blood collection on those HCAP people at the same time, and these markers will be collected. So you know, HCAP only has 2 waves in the field right now, 2016 and 2022. The next one will be in 2026. So it's just going to take a little time to get there. But eventually, you know, that something like that would be possible. Okay.
And do you know when HCAP 2022 will be publicly available?
I do not. That's another arm of the study.
Okay, great. So I think we have mostly worked through. Oh, you could close off with this basically. it's a pretty broad question, but what are the next steps for use of these markers in HRS? It's broad, but it might be a good way to close.
Well, like I said, again, I think we had enough evidence here to consider them strong enough biomarkers to keep longitudinally. So as I've mentioned, we will have these measured again in 22 at least, and again in 2026. I think for us, what we're really interested right now, I kind of hinted at it before, is harmonization. So we did this using the Quantrix platform, but there are other platforms out there and they don't line up perfectly. So what we're hoping to do is understand more how the platforms relate to each other and how we could crosswalk with other studies that maybe have used other platforms, so that we could pool data, or at least people could more easily do cross-study comparisons. So that's sort of on our horizon right now. And as always, we try to keep up to date with sort of newer developments. I would say, so one thing we're considering is whether we would move or add another p-tau measure onto this. So p-tau 217 in particular. It remains to be seen. But we move where the science moves. So we'll make that decision in the future. But again, we have a lot of banked samples. So if a new marker comes along that is really promising, and we think that we can do it from a previously frozen sample, we can go thaw that out of the freezer and actually sort of have longitudinal data right at hand. So with that repository, we have a lot of possibilities.
Perfect. Well, thank you very much.
Thank you.
It is 12:59. That was fantastic.
Perfect timing.
Folks, again, be on the lookout. This webinar will be posted on the HRS website and also on the EdDem website as well, relatively shortly, as well as will the additional questions that Jessica wanted to follow up on that weren't fully answered from the Q&A. So thanks again, everyone, and I hope everyone has a great afternoon.
Great, thank you.